Thank you all for joining. Hello and welcome to my talk about decision-ready data in biologics R&D. So, my name's Phil Burke, and I'm a technical account manager here at LabKey, and I primarily work on our biologics LIMS system. So, a little bit about me. Um, I'm a technical account manager here. My background's actually in molecular biology. So I did my PhD at the University of Washington, working at the Fred Hutchinson Cancer Center in the Subramanian lab. And there we primarily studied the sort of fundamental process of mRNA translation. Um, so in this case, translating mRNA and producing proteins. And what we were really interested in was how mRNA sequence impacts protein synthesis, like the amount of protein output that you get. Um, so a big part of my PhD was building high throughput sequencing-based assays to try to determine the effects of mRNA sequence on mRNA stability and protein synthesis. After I finished my PhD, my background in kind of high throughput sequencing technology led me to a role at a small company in downtown Seattle called A-Alpha Bio. And this is a biotech that basically uses a high throughput yeast display measurement to measure protein-protein interactions in high throughput. Um, and so we primarily took these sort of large scale measurements of protein-protein interactions and applied them to antibody characterization and machine learning problems. So we generate really large data sets of antibodies and antigen interactions and then use those to train ML models. Throughout this process, I gained a lot of experience with basically the biologics art research and development process. But I also learned a lot about the best practices and common pain points of managing both samples and data throughout the R&D lifecycle. And that ultimately led me to working here at LabKey. I found that kind of more project management and LIMS system development actually suited me pretty well. So here I'm working on our Biologics Laboratory Information Management System, or Biologics LIMS, which is really built to just handle data of all sorts. A little bit more about LabKey. So, LabKey is a software company that really specializes in the biologics life sciences. We've been around since 2003. And since then, we've worked with academia, pharma, biotech, and government research. And one of the things that is sort of foundational for LabKey is that we really work back and forth with our clients. So, as basically, we work directly with our users to ensure that we're building software that works for their needs. And because so many of our clients have focused on building biological therapeutics, one of the outcomes of these back and forths of the last 20 years is that we've basically ended up building a LIMS system that's really purpose-built to handle molecule discovery and development research. Um, so one of my goals as an account manager at LabKey is just to use what I know about biologics data management to try to help folks in the industry optimize how they're managing their data and their reporting practices. Because while, data management isn't exactly something we think about on a day-to-day basis, maybe it is for you. I guess it is for me. It's certainly something that matters a lot. Um, so making the right decisions in biologics research is critical. And this is where data management becomes really important. So you might be saying, well, why? I mean, making the right decisions is kind of antithetical to science, right? The reason we're doing experiments is because we don't actually know what's going to work. But in the biologic space, when you're building biologics therapeutics, it's actually very important to make the right decisions whenever possible because biologic therapeutics are really time and resource intensive. So you can't afford to be wrong that many times. And while we can never fully remove uncertainty, we should still try to make the best possible decisions based on all of the available data. Um, so that kind of leads me to my sort of thoughts on some of the challenges that impede decision making. So a few of the challenges that I can think of that actually cause it to, be more difficult to make the right call when you're trying to pick a therapeutic lead or, do a follow-up experiment are these are some of kind of the big categories, but just not being able to find your data or having incomplete metadata, data context, and then lag in both, input lag, so data logging, and also sort of data handoffs between teams. Like, oh, we need this design so that we can move this step of the experiment or of the project forward. Um, and then that also ties in with another kind of typical issue of just navigating many disjointed systems. So, working across many different types of software to try to, keep things organized. It can get a little bit out of hand. And finally, sort of the most important part is that we as scientists probably spend quite a bit of our time carefully explaining our experiments and results to others. And often that manifests in saying, oh man, I know we have a slide deck on this somewhere. So that's kind of the point here is that nobody's perfect. We've all probably found ourselves saying some version of these phrases throughout the research process. And a lot of this friction really just comes down to a lack of data visibility, and a lack of kind of structured cross-team communication. So when we think about sort of the archetypal biologics R&D team, you've got a lab operations group, which basically is doing your equipment, your automation, your reagents. You probably have a well, I hope you have a research and development group that's doing hypothesis generation and assay design and execution. A data science group that's probably doing more of the sort of high level data analysis, data organization, maybe making some of the plots and scripts that you're using. And then you've got a leadership group. And here I'm thinking kind of more of your PI or your CSO. So somebody who is basically making the final call on what project directions you should be pursuing, and someone who's carrying the weight of making, the financial decisions. And while it's easy to really think of these sort of four pillars as completely independent units, we know that's not the case. For example, your R&D group is probably involved in basically every step of this sort of overall R&D, scientific process. Like they're going to be helping lab ops, with equipment. They're going to be using those reagents and stuff like that. They're probably doing some of the data analysis and the slide deck building and results reporting. And I hope they're involved in determining what your project direction is. I hope there's a good back and forth between the CSO and your R&D team. And this is true for the other teams as well. Of course, data science is probably helping out with instrument automation and like is probably helping with hypothesis generation and design and stuff like that. And then I would imagine that leadership is also, working closely with R&D to come up with hypotheses and assays and things that you want to test. So once you start getting into these back and forths, there are just a lot of different people who have very different responsibilities that need to be able to communicate with each other and keep track of what's going on. Um, and so a relatively simple example of sort of a communications workflow that happens is just basically going from an in silico design. So, hey, we've designed this antibody, we think it's a good idea, to building therapeutic leads. And so this is just sort of the basic process throughout which you would do that, where your data science team or your computational design team comes up with some molecules and they want to test them. So they talk to the R&D scientists about, oh, hey, we should clone these. And R&D scientists go back and forth on the cloning design. And then maybe they talk to a different group to do the protein purification and characterization, back and forth here. And then maybe it's a completely different group of researchers who are doing functional testing, like ELISA assays and things like that, to determine the actual, effects of these molecules that you're building. And then eventually you've got a big package of data. You go back to data science. Both of you start digging into it together. Data science maybe digs into it even further, uses it to develop new models. And really, each of these steps of these process, there's this back and forth communication that needs to happen, and this can really slow down your development process unless you've got a system in place for your users to rapidly assess and review what's going on in the project. Like, what's the status of this cloning? What are the outcomes of this ELISA experiment that you did? Um, and all of this really becomes very important sort of at this last stage where you're selecting hits and leads to prioritize, because your leadership team also needs visibility on this whole R&D process to make the correct decision. Like this is this the molecule that we want to go forward with? So my little example of that here is, let's say your CSO has seen this whole package of data and now they need to decide on, okay, are we going to go with molecule A or molecule B? And so the CSO goes and they ask the scientist, well, hey, like, I think molecule A looks a little bit better. Maybe we should go with A. What do you think? And you as a scientist are like, wow, there's something about molecule A that just does not sit right with me. So you really want to be able to call up the underlying data that you have on molecule A and see what it is that's setting off warning bells in your brain. And sure enough, 3 years ago, you tested molecule A, for a completely different project. And while it had a very promising therapeutic profile, it had really bad expression. So you didn't pursue it over leads, X and Y. And so this is the kind of thing that these are data that, are associated with this project that you really just need to be able to access and compare. You want to be able to compare between A and B, but you also want to be able to compare between A, X, and Y from 3 years ago. And there are lots of different factors that, could be different between this current campaign and this old campaign. Like maybe, you were using a slightly different sort of sequence variation, or maybe a different expression vector or cell lines or things like that. And it could be that, you could make some tweaks to molecule A and optimize the expression and actually produce a really good molecule. It might be worth it. The trick is this information can kind of get lost over time, and you just really need a means of keeping track of these decisions that you're making and why, so you can actually choose and be really confident in going forward with molecule A. Um, and this type of kind of historical data tracking and just keeping track of data associated with your samples in general gets even more difficult when you add a third party into your mix. So another example is, let's say you've decided that you're going to go forward with molecule A and you're going to go do an external screening campaign with this and you're going to work with a CRO. Well, now you've introduced this third party and things kind of get even more sort of difficult to keep track of. So as an R&D person, you're sort of the internal point person. You're going to communicate some designs, maybe some initial results, and then work with your lab ops team to send physical samples to this CRO so that they can kind of follow the work plan that you've laid out for them. And then some amount of time passes, and then eventually the CRO sends you back usually an email with like an attached PDF or maybe some CSVs or maybe a slide deck. But basically, certainly not structured data, or very, very likely not structured data. And so it's usually around this point, 2 to 4 weeks in, it's been about a month, that your CSO comes up to you and says, well, we paid a lot of money for this CRO. Like, what's the progress on that screening campaign? I'm so curious. Like, and you don't want to have to say, oh yeah, that data is in an email. I'll forward it to you, and then like we can go through the slide deck together. Like nobody really wants that. What we want is transparency. We want to be able to sort of access the data and self-serve. This goes both ways. Like if you're a CRO, you definitely want your clients to know exactly what it is you've done. You don't necessarily want like your CSO to be left in the dark. You're doing all this good work and sending people stuff. You want them to know that you've done it. So ideally, the goal here should be to collate these results in a structured and visible manner and be able to sort of track and associate all of these results with the samples that you have in the lab and the samples that you've sent out. And really, I mean, this is just good policy in general. You want to do this for all of your samples in your data. The trick becomes just making this an easy thing to do. So with that, these are sort of the three key capabilities that I think are important for solving these problems in getting your data ready to sort of make decisions. The first is building out structured data architecture. The second is building out workflows for your operations that you kind of keep track of different stages. And the third is making it really easy to ingest your data, either by automated capture or just by having sort of software setup that allows you to really easily log things. So structured data architecture, this is basically the goal in a biologics workflow should be to unify entities like sequences, protein sequences, molecules, and stuff like that with physical samples. So you have 5 mgs of this protein in a tube somewhere, and then assay results. You use those 5 mgs of proteins and you ran them through flow, and this was the binding. And then the results as well, like, and you found that it bound to this antigen at a much higher rate than, the wild type version of this antibody. These should all be tracked in the same relational system, if possible. Um, so in this case here, what I'm kind of showing is just, yeah, we've got, this antibody Herceptin, and it's got these sequences that build it. It, makes this Fab. We've expressed it in this cell line. It produced these samples. We used this sample in some assay down the line, and then produced data related to that. So this is sort of just a sort of schematic of what this could potentially look like. And then the other kind of huge benefit of having structured and centralized data is that if you build your data to be really structured and queryable and metadata rich, you actually just directly enable some really powerful things. You can enable cross-study comparisons. So it's easy to say, oh, like how was molecule A this week versus 3 years ago? Like, let's compare those data. And trend analysis. So like over time, like this thing has been getting better. Oh, this lot of reagents has been producing worse and worse results. And then most importantly, perhaps in 2026, is you also enable ML-driven modeling and AI-based analytics approaches. And so utilizing your data, really leveraging your data in AI-based sort of analysis is something that I think a lot of people are interested in. And it's really honestly, it's just a good thing to be doing. You have your data, you should be getting the most out of it. So trying to use it to dig into any sort of ML based approach is just, I think, something worth doing. Um, the next kind of one that I mentioned was building out operationalized workflows. And this is really just building workflows that you can have seamless experimental handoffs to keep things moving. So if you've got repeatable lab templates that let you do handoffs through experimental stages, ideally integrated with your ELN. So you've got a lab notebook where, say, you're doing, oh, well, we've designed some molecules, we've got notes related to those designs. We're going to hand that design off to R&D. They're going to look over our notes. They're going to say, okay, yeah, we'll do it in this vector. That looks great. Then they're going to sort of move towards cloning. Maybe they'll order they'll order the plasmids, and then they'll hand that off to your lab ops team to actually do the physical cloning steps. Ideally, you just want people to be able to sort of document each of these handoffs and know that the project is at a given stage so that they can continue to move things forward in a really really quick way. It's also, I think, very just important for teams to have a single source of truth. You want everybody to be operating on the same version record, so that you reduce sort of having to repeat the explanation. Oh, like what's going on here, or potentially even rework. Like we did this experiment before, but somebody else didn't know about it, and they did it again. Um, and there are some clear benefits to kind of building out workflows as well. So as I mentioned, yeah, it keeps things moving forward, quickly. So you don't need this back and forth communication to move to the next step of each stage of the experiment. But also building out standardized experimental templates does give you a basis for both quickly initiating new work. So it's like, oh, we want to do this experiment, bam, let's do it. You don't need to spend a lot of time building a new lab notebook and thinking too hard about it. And they can be really helpful as your group expands. So if you're adding new people, having these templates built out makes it really easy for your new researchers to come in and say, oh, well, I'm doing this experiment and it looks like I follow these steps, and somebody else, walks them through it the first time, but then they're ready to go. And also, as your company expands, doing these types of things can also give you a built-in audit trail, which can be a real benefit if you ultimately need to be following sort of federal guidelines like compliance guidelines. Um, so those are some of the reasons to build out workflows. And then the last thing that I kind of wanted to talk about was this automated data ingestion and structured capture. So the ultimate goal here is to make logging your data as easy as possible. And in this example workflow, I've got a technician or someone working at, basically a core facility is running Illumina sequencing on your sample. They're operating the sequencing machine. Rather than those raw FASTQ files, basically going back to the scientist and then them having to do a bunch of manipulations in R, Python, I'd argue that the ideal path is a series of sort of automated uploads and transformations where the data first goes to the cloud, and then you have some automated scripts that basically see, oh, hey, data is here. That's great. And this data is then taken and it's run through a series of sort of analysis transformation pipelines where you go from reads to like counts of reads to summary data to plots. And then these output plots saying, hey, this is the QC of your sequencing run. These are some of the top results. This is, your hits arrayed from, best to worst. All of those just go right back to the scientist and they can look at these summary plots. And there was no real time wasted or spent within any of this. You just have this logged structured data that's ready to go. And so building these types of extract, transform, load operations, basically it allows your team to very quickly and directly access results. And it also importantly eliminates any variations that might arise from manual data input and processing. Of course, even if you don't want to do this kind of fully automated stuff, you can still gain similar benefits by just having a user interface that makes logging very structured and convenient. So, like, the sort of ideal here is that a researcher finishes an experiment, and then than taking that data and going and analyzing it on their own local machine in Excel or cleaning it and making some plots in R, you want the easiest route for them to be simply uploading this raw data file into your data capture system. And you've configured that system to then clean up the formatting of the raw file so that you just get your data in a nice structured way. After which point you could do your filters or comparisons. You can select your top hits. Maybe you can make standard plots, all just within that user interface. So that's the ultimate goal for this. Um, And with that, so these are, again, the main capabilities that I think are crucial for effectively managing data throughout the biologics research and development process. If you're interested in establishing this type of system in your own lab, I would encourage you to check out LabKey Biologics LIMS software. Our system is basically purpose-built to model biologics R&D relationships between sequences and samples and experiments. We've got a built-in ELN and workflow tools for managing kind of complex laboratory workflows. And we're really focused on integrating external tools and doing basically easy data capture, making it easy to upload raw data files into a structured, searchable, decision-ready data package. And so with that, I'll stop talking. If you want to kind of come talk to us, discuss how some of these capabilities, might actually be applied to your workflows, um, we would absolutely, be interested to talk. We run these free guided evaluations where we meet with you, we learn about your data, we think about how it might be applied to this type of system, and learn a bit about your use case, and then we offer kind of free demo trials where we build a demo around your data and show you what it would look like within the LabKey system. And aside from that, keep your eye out for your email and recordings, and we'll send you this recording and a demo link, of course. And yeah, just thank you for joining. Really happy to take questions on this. Anything anyone wants to ask, or we're here for it. And of course, you can always email us as well.